All right, can you hear me?
Yes.
Good, we ready to go? So, great. Thank you guys for turning this into an online session. It's been super fascinating to hear about everybody's work. So many of us are interested in social interactions and social life. And I myself come from a more evolutionary direction, and in particular, how evolution manages to engineer social life.
So
how
you have a weird screen that said build order.
Oh no, okay.
Yeah.
How about this?
That looks
How
good.
about this? Do you see this?
Perfect, yes, perfect. Good. Go ahead.
Okay, here, let me just fix one thing over here. Okay, and boom. Good? Better?
Yes, much better, thank you.
Okay, so how does evolution engineer social life? How does evolution build a system where we can see multiple individuals interacting dynamically and making decisions over a distributed network? And today I'm going to tell you guys about one such system, in particular using social insects and socially exchanged fluids. Do you guys see me or no? I'm there. Do you see my video? Yes? No?
We do see you. We can see you.
Okay. All right. There. I fixed my situation. Good. So social insects and socially exchanged fluids. So ants. Ants are already well known for their ability to make complex decisions and solve complex problems. For example, finding the shortest path between 2 points, exploiting and finding the best resources, and doing this in the optimal way. And even they're excellent at finding the best nest sites. If you destroy their old nest, they can choose between different nest sites and choose the best one. But fundamentally, each of these decisions are short-term, acute responses to specific situations. How do they make longer-term decisions? What do I mean by a longer-term decision for a social insect? How do you transition from something like this, a single queen, into this, a fully mature colony of thousands of individuals, all without central control, all in a distributed way, and including a high amount of division of labor? How does such a colony of thousands of individuals shift their priorities from focusing on growth of that colony into prioritizing reproduction and dispersal? One way to think of this is the analogy of a company. The first months of a startup, the types of workers you want working in that startup and the type of organization you have are going to be vastly different than the type of organization and the type of individuals working there once that company has grown into a giant multinational corporation. When does it produce spin-offs? Who decides? So where in biology can we find these kinds of answers? And the place that I tend to look is at socially exchanged fluids. So social fluid exchanges tend to only occur in really important biological behaviors, things like mating and parental care. And these are situations where an important cargo needs to be transmitted from a donor to a receiver over a private communication channel. And often over this private communication channel, there are other more interesting things that are also transmitted as well that can be a bit more manipulative or more directive in nature. So one great example is in Drosophila seminal fluid, where along with his genetic material, the male transmits to the female a whole bunch of different things that can influence her behavior, her fertility, and aspects of her physiology. So one of the reasons I think social fluid exchanges are particularly interesting for collective behaviors is because they provide a private communication channel for consensus building, which is a critical thing in most collective decisions. And in particular, the social fluid exchange that I study is the one down here in this quadrant, trophallaxis. So this is a beautiful and quirky social fluid exchange that occurs in many but not all social insects. It's generally considered a food transfer. And as you can see between these ants, one is regurgitating a little green liquid from her crop, her social stomach, and she's giving it to another individual who is then drinking it. And for species that engage in this behavior very, very often, each individual in the colony is connected through this network of fluid exchange. So we can think of trophylactic fluid, the fluid that's transmitted, as a kind of social circulatory system that transmits nutrients and signals to each individual in the colony, just like our circulatory system transmits nutrients and signals to each cell in our bodies. So in my first postdoc in Lausanne, I decided to figure out what is being transmitted in this fluid. And so I used a lot of different methods, mass spectrometry and small RNA sequencing to find many things are transmitted. Nestmate recognition cues, which are called cuticular hydrocarbons for those who care about that, small RNAs, many different types of proteins, and an important insect developmental regulator that's called juvenile hormone. So a whole bunch of things are being transmitted. And we can think about each of these different components as different forms of information that are being produced by individuals and then being let to diffuse over this social network, okay? And for example, talking about these many different proteins, they're a very rich form of information. So what are proteins, for those of you who are deeply physicists? Proteins are the cogs, nuts, and bolts that make up the machines that are living matter. So I'm made of proteins, plants are made of proteins, ants are made of proteins. And proteins are built based on specific stretches of DNA, which are genes, and they are produced for specific times and specific places in the development of an organism. And so here in trophylactic fluid, we find about 40 different proteins that are secreted into this fluid compartment that is shared over the colony. What are they? So I'm not going to go deeply into this, but this is something that we've looked at in a few different species of ants and also in honeybees, and then another group looked at in one species of termite. And across all these species, we find more or less the same groups of proteins. We find proteins that are involved in defense. This is not surprising because fundamentally we're looking at the contents of the gut. So we find, did I say defense? I meant digestion. So we find proteins involved in digestion, and that is not surprising. We also find proteins involved in defense, immune defense. This also is not so surprising, but a bit more interesting in that while the gut is one of the first lines of defense for an organism against pathogens, it's also very important in individual organisms that live in close contact with one another, as we are seeing with the coronavirus. Why are we not at this conference together? Because of the risk of transmission. And so social insects are actually a really interesting model for studying social immunity and epidemiology, because they may have ways to prevent one another from getting sick. But fundamentally, the real thing that caught our attention was the presence of many proteins that were involved in growth and development. Why are they there? Adult ants are fully grown. They don't need to grow anymore. They're not like Yasmin's beautiful plants that are continually growing. An adult ant is fixed. So where are they aimed at? Where are these growth and development proteins focused on? And so for that, we have to think a little bit about the social insect colony. Often when we consider a social insect colony, like bumblebees, we often think about nurses and foragers, these two interconnected groups in the colony that have their social interaction network. But in addition, there is a third important group, and that is the brood, the young, the larvae. And in fact, the larvae are also in part of this trophylactic network. So as you can see here in this beautiful video of a hornet feeding her her larva, or the larva feeding her adult, which the trophallaxis can go both ways. So, they're also part of this network, and that leads me to a hypothesis that a lot of my lab centers on, which is that trophallaxis is a means to socially manipulate development. So the idea here is that the workers within the colony can act as sensors for the colony's social environment. Each individual is a sensor for the social environment. And they then translate their response to the social environment into the molecular components of their trophylactic fluid. So turning up this dial, turning down this dial. And then these components, when fed to larvae and filtered through the social network, they can influence the developmental outcomes of those larvae. Then these larvae grow up, they then become adults and can feed back onto the social environment. So this model is quite cool because it provides a mechanism for what we've observed in social insect colonies for hundreds of years, if not thousands. The fact that they're able to self-regulate their colony development without central control. So is this true? Is there support for this hypothesis? So to start diving into that, first question is, can trophylactic fluid components actually influence development? And so I mentioned that I had found this juvenile hormone, this important insect developmental regulator, is present in this socially exchanged fluid. And so this was kind of novel because normally this developmental regulator, juvenile hormone, is present in all insects, many invertebrates, but circulating in their own bloodstream. So fruit flies have it, beetles have it, but it's not transmitted. Here we found it in a transmitted social fluid at reasonable quantities, approximately similar to that in hemolymph. So we wanted to see if it could have an effect on development. And so to look at that, we took groups of ants and then we gave them larvae to rear. And we supplemented their food with either solvent alone or solvent plus this juvenile hormone. And then over time, we looked at the pupae and saw what they became. And what we found was that the adults that were reared in the juvenile hormone supplemented condition were slightly bigger than in the solvent-only condition. But what was more striking was more than double the number of individuals were reared to adulthood in the juvenile hormone supplemented condition. So another way to approach this is that the same number of adults that are rearing the same number of larvae produce double the number of new adults when they have high amounts of this juvenile hormone in their trophylactic fluid. So what we can then imagine, this has lots of interesting implications. So it sort of suggests a form of quantitative voting that is happening over this social circulatory system. So if I want more individuals in the next generation, then I produce more juvenile hormone and send it out over the social network. Also, it means that individuals who are more closely implicated with the rearing of the brood probably have a greater say in what those brood members, what those larvae become. So that's some interesting implications there. So yes, we do see that developmental outcomes can be influenced by components of this fluid. But do we see that this fluid can be adjusted by the social environment? So to look at that, I took individuals from within a colony. I collected their trophylactic fluid, and then I isolated them away from the colony for 14 days and collected their trophylactic fluid again. And then I compared what they have in there by proteomics, which is looking at the abundance and which proteins are present in this fluid. And so this is what you see here. You see by the rows, these are different proteins. The columns are different samples. And you have it's a heat map, and so abundance is shown in color. And so what you can see is that many different things change in abundance. In fact, 19 different proteins change significantly in abundance. So yes, we do see that social environment can influence the trophylactic fluid composition. And in addition, of those 19 proteins, 4 of them happen to be from the same family of enzymes, which are called juvenile hormone esterases. They break down this hormone that I mentioned in the previous chunk of the talk. So not only is the molecular composition of this fluid changing with the social environment, but it's changing in a way that's specifically surrounding these growth and development related proteins. So yes, cool. It looks like social environment can indeed manipulate the trophylactic fluid. Now, we haven't yet gotten to the dotted arrow, but one cool thing we can study, because we're working with proteins, is how evolution has actually built the system, how it has been built over evolutionary time. Because we can go into the genomes of many other species and take a look at what's happened. So I'm going to specifically go a little bit into this phylogenetic thing. I'm going to try to keep it simple and not too jargony for you. So in particular, I'm looking at this family of juvenile hormone esterase-like proteins that we find there that can break down the juvenile hormone. And just to give you a bit of context, typically we have one of these per species. Even though there are many different enzymes that do things like this, there's usually only one special one that actually does the breaking down of the hormone. And this is defined by its specificity for that hormone and also its sort of expression pattern, which is sort of where you find it in the organism. There's been only one such enzyme that has been characterized and found for a social insect, and that's for the honeybee, Apis mellifera. And here, in the species that I'm working with, primarily carpenter ants, we find a whole bunch of these, but they're present in this socially exchanged fluid rather than in the insect bloodstream, the hemolymph. So to explore what's going on here, we reannotated all of these types of enzymes in this main species we're working with, this Florida carpenter ant. And we found 27 of them. And they're of different quality, and this depends on how the genome was done. It's not so relevant. But one thing we did find was that they're arranged in what are called tandem arrays. So one of the ways that evolution is tinkering here is by copying and pasting. And what you find when it does copy paste is you find many different genes in sequence in the genome that are slightly different. And so we find a few different arrays of this in the genome here. In addition, one handy thing we have as well, and so these different arrays tend to fall into these 3 different blocks. These 2 are actually probably connected arrays that we find present mostly in trophylactic fluid, and then some a bit in hemolymph and some a bit in larvae. And a handy thing that we have here, looking at this family of proteins, because they're targets for pesticides, someone's already done a crystal structure. And so we know where the business end of this protein is, and what are the specific amino acids that are used to actually do the cutting of the juvenile hormone itself. And so we can look at whether those are intact in the proteins. And we can see that for specifically the proteins that we find in this fluid, all of those proteins are intact and should be able to do this reaction, and not necessarily the other 2 groups. So we wanted to see, is this different in other species? Is it the same? What's going on? So looking at another carpenter ant, at a more distant ant from a different subfamily, and at the honeybee, we can see that in the 2 types of carpenter ants, 18% of all of the protein that these ants are putting into this fluid is just this family of proteins. 18%. That's a huge amount. I mean, you'd think that it's your stomach, you're going to mostly secrete digestion enzymes, but here they're putting a whole bunch of this growth and development stuff in there, which is pretty crazy. We don't necessarily find that, though, in the other 2 species that we looked at. Similarly, is juvenile hormone there or no? And we find the same pattern. So it looks like this is an evolutionary adaptation. This is something that evolution has built specifically between these other subfamily of ants and the ones that we've been focusing on. So we can dive in a little deeper and see what's happened. So we looked deeper into more genomes, looking a bit at this other subgroup, the myrmecine ants, mostly at the formicine ants, and using another genus that's very successful, Formica, to have a comparison for our genus of Camponotus or carpenter ants. And we have proteomes for some of these. And then we made a big tree of all of these different genes. And what you can see in yellow is the subtree where we find the trophylactic fluid proteins. And at the base of that, we have the juvenile hormone esterase that was characterized in the honeybee. So we know we're looking in the right place. So if we want to just look at that yellow portion, that's what you see here. And what I want you to focus on is the difference between the yellow and the orange. So the orange corresponds to those 8 species of Formica, and the yellow corresponds to just 6 species of Camponotus. So what has happened is that somewhere in the evolution of Camponotus, they have done a lot of copying and pasting. So they've built up 46 different versions of these things over those 6 species versus the 8 or the 10 in just 8 species. So we've seen a lot of copying and pasting. And then if we dive in a little further and we look at, we overlay the results that we have from proteomic information, so we can see how abundant these things actually are in this fluid. You can see that as the tree goes from the inside out, that's evolutionary distance. So the further out, more evolutionary changes have happened. And so you can see that with more evolutionary distance, we are finding more abundance of these trophylactic fluid proteins. So they're becoming more abundant in this fluid. And all the ones in gray are in species where we haven't actually done proteomics. So we only have the ones that are in color and black. So that's quite cool that we're seeing this shift into this fluid. And then lastly, for the aficionados, we are, you can also measure whether there has been what's called positive selection, which is essentially a push, an adaptation in a particular direction. And we see that there are branches of the tree that have significant levels of positive selection, specifically leading to these highly abundant esterases. So what we're seeing here is a regulatory neofunctionalization. So a new function is being built by differential regulation. And we're seeing that because we see duplications, this copying and pasting. We're seeing positive selection, and we're seeing a change in localization. Now, one question is, are we seeing also a change in function of these things? How is this working? And so one way to look at that is by using an inhibitor of this family of enzymes. So this OTFP is an inhibitor of this family of enzymes. So when we inhibit the enzyme that breaks down the juvenile hormone, we should have an increase in the juvenile hormone. So again, we took the same approach of giving groups of ants larvae to rear and supplementing their food with different components so that we can see how they influence the larvae when fed by trophallaxis. And so as I mentioned before, when we increase the amount of juvenile hormone in the trophylactic fluid, we see an increase in the number of larvae that are reared to adulthood. Now, we also have juvenile hormone esterases in the same compartment that should be trying to break down that juvenile hormone. Now, break down the juvenile hormone. Now, if we inhibit those with the OTFP, then we should see an increase in juvenile hormone in the number of larvae reared to adulthood. And in fact, that is what we see. So when we have this inhibitor, we see a similar response to when we have added juvenile hormone. And when we have them both there, we see an even greater response where we have more individuals reared to adulthood. So that's very cool. And what it tells us is that fundamentally, how does evolution engineer social life? And we're seeing one way that it's doing it. It's doing it by taking within individual tools like this juvenile hormone and also this esterase, that are normally circulating in a solitary insect's bloodstream. And here, instead of having it just circulate in its own bloodstream, it's putting it into a social fluid and transmitting it out over the social network to communicate with other individuals in this distributed network of individuals. And it's doing this, it's doing that by duplicating, repurposing, and then tinkering. Yeah. So this is bringing together these two nice big questions, how to make long-term collective decisions and how to engineer social life. And it's doing this with this superorganism's circulatory system. And now, because I haven't heard anything about being end of my time, I'm going to tell you a little more about how trophylactic
about
feeding,
4 minutes.
How trophylactic feeding can mechanistically alter development. So how does this actually work? And so this is some work that I was doing in Ofer Freinemann's lab at the Weizmann. And so this is a quantitative tracking of larval feeding and their concomitant growth. So what we were doing was feeding young queens a fluorescent food source, which they then feed to their larvae. And so we can watch the larvae get fed over time and see how they grow. Now, there are a lot of tricky things about doing this. Fundamentally, you know, the larvae are in close proximity to each other. They obscure each other. You have nasty collisions. In addition, they are of very variable fluorescence. They also grow over time, and so you don't have, and they are very different sizes, as you see in the upper picture. And also the body posture changes. So we have a lot of sort of computer vision and identity tracking problems, but we've managed to put something together. And so you can see these lovely queens feeding their babies right here. And these are 2-week experiments, about 1 frame every 30 seconds. And as you see here, we're managing recognitions not of every single frame of all larvae because of the collisions, but we're managing a number of them. And it seems to be enough to get what we need. And fundamentally, we're doing this with a combination of a software called FimTrack, which was developed by Benjamin Rees, and the algorithm DBScan, which I'm using to cluster the segmentations and then do assignments for larval ID. So for now, I call it very supervised machine learning. And so what we can get out of it is a view into the entropy within parental care. So how do we see favoritism? How consistent are the queens with their young? The hunt for nepotism in social insects is a long one, and so we're finally going to be able to see a little bit of a view into that. So what you see here at the bottom is about 1 week of an experiment of a queen who is caring for 3 of her larvae. And you have larval area in the first plot and the amount of food consumed as measured by total fluorescence in the second plot. And I'm going to point out a few things. So here we can see that the red larva was eaten. So there's a lot of cannibalism in social insects that people don't like to tell you. But yes, they do eat each other. It's reabsorbing the nutrients into the the superorganism. And in addition, we can see many different specific feeding events. The most clean one is the very first down at the bottom. We also have a nice one a bit further up. And so we can individually see when each larva is fed, and we can see how they are growing over time. So more results. This is not being submitted next week, so hopefully you'll see it in some time, hopefully by the end of the year. But in general, we can explore further how these components are being transmitted over the social circulatory system and how they're being used to make complex collective decisions. So with that, I want to thank my young lab at the University of Fribourg. We only got started less than a year ago. And also Ofer Freinemann's lab at the Weizmann. And then my collaborators on the positive selection work, Eyal Prüfman at Haifa, and then the place where I started this whole trophallaxis exploration, which was in Richard Benton and Laurent Keller's lab at the University of Lausanne. So thank you all very much, and I'm very happy to take questions.
Thank you so much. Okay, so if anybody has questions, please raise your hand. Okay, we have one from Grace.
Hi, I was wondering if during the experiment where you were measuring the proteins in the trophylactic fluid, did you do variations on that, like where you maybe removed ants from the hive and then, sorry, colony, and then looked at what happened to the remaining ants, or isolated them with other members of their same caste or something like that?
So we're doing actually quite a lot of different permutations of this. Now, there are routine differences between nurses and foragers in the protein abundance when they're trophylactic fluid. So I can tell you that they correspond to expression, differences in expression, protein expression as well. One tricky thing is how believable are separations in lab colonies. I've just finished about 2 weeks of field work with fully established colonies out in Florida to see the real thing in the flesh and collect the fluids from them out there. So hopefully I'll be able to tell you more in the future.
Okay, we have another question from Hantang.
Hi.
Hi.
It's just a very quick question. I just feel like I have to ask because I see my favorite species fire ants on there with the negative.
Mm-hmm.
Feels like having a kid like scoring very badly in the exam. So does that mean that some of the species in ants or in other social insects doesn't have this growth control mechanism?
They have different ones. So I can tell you that Solenopsis invicta, they pass around vitellogenin. They also pass around juvenile hormone binding protein. And also they pass around the OBP, the GP9, by the way, in case you're part of that world with the polygyne monogyne. So so they pass around different things that are also involved in growth and development, but they're probably different systems. So honeybees also pass around growth and development things. Most of what we know that's in royal jelly is produced by the same glands that produce the proteins in trophylactic fluid. And when I measure the trophylactic fluid of honeybees, you find many of the same proteins as you find in royal jelly. So many of which are also involved in growth and development.
Gotcha. Thank you.
Okay, we have a question from Greg.
Oh, can I just add one little finish there?
Yep.
In terms of other social insects, so there are not all social insects engage in trophallaxis. So we're also doing a big evolutionary study now, doing comparative work with species that don't do trophallaxis, because we would imagine they won't have these growth and development things in their trophylactic fluid. Okay, Greg?
Yeah, I was curious, totally interesting talk. And I was wondering if can you relate, controlled perturbations that you do to the colony to what's going on in the trophallaxis, or the frequency, or how tight is that connection?
I would love to know. So now, so I mean, there have been a lot of strides in the recognition of trophallaxis behavior since I got started on this work. So for example, Tim Gernat in Gene Robinson's work in Gene Robinson's lab has done some nice work recognizing trophallaxis events in honeybees. And the network that they trained for that has worked in ants. So a friend of mine in Ulfer Feinerman's group has tried it with ants, and it worked pretty well there. So, we do know some behaviors just from like ethology that do lead to more or less trophallaxis. So one such behavior is when you socially isolate ants and then feed them and then reunite them, they do a lot of trophallaxis. So that's one major one. In some species, you see aggression-related trophallaxis, like, here, give me your lunch money. Okay, and then they regurgitate. So, yeah, so there are a number of situations where you see it. I have done some work that I haven't published on the network of trophallaxis around the queen.
Mhm.
And you see things that don't entirely, like for most trophylactic interactions, they have to do with spatial structure and the normal social network. But with the queen, there are some other interactions that occur, and it might have to do with diffusion of components that she has, but we don't know.
Thank you.
Okay, thank you so much, Adria, for this beautiful talk. We're going to move to our last speaker. Her name is Sang Choi, and she's actually going to talk about the, I think, trophallaxis behavior that was just mentioned at Jean Robinson's lab.
Hello, can you hear me and see the slides?
Yes, yeah, we can hear you.
Yeah,